Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. Good evening to all. I hope I am audible to you. So, what we have seen yesterday, I would like to revise, and then we'll see the next portion of blood and its components. So yesterday, as I told you, the blood and its component, very basic chapter from your anatomy physiology. And if you understand this blood chapter, once you understand this blood, components of blood cells, their hormones which control the blood cell production, and once you understand the plasma proteins, component of plasma, blood clotting factors, the process of blood coagulation, right? So if you understand this blood and related components from anatomy physiology point of view, so you will understand the anticoagulants, anticoagulants, thrombolytics, hematinics. So there are 3 chapters which are based upon the blood chapter from pharmacology. And you know that anatomy physiology is a basic subject, and once you understand HAP, biochemistry, microbiology, then only you are going to understand the pharmacology. So that's why we are taking blood first in a unique way. I used to teach for GPAT Niper DI Pharmacist. So that is known as a interdependent subjects, right? So blood is a basic chapter, and based upon the blood you have anticoagulant, coagulant, thrombolytics and hematinics chapters in the pharmacology. That's why we are discussing the blood first, and then we'll see the drug acting on a blood from pharmacology, right? So that's why, right? Yesterday we have seen what do you mean by blood, what are the components of blood, what are the plasma, what are the cell fraction. So plasma, cell fraction we have seen yesterday, right? So we have also seen what are the component of plasma. Okay, what are the functions of blood? There are several functions. We have also seen some normal values like glucose, creatinine, cholesterol. These 3 are important. Glucose 100 to 120 milligram per deciliter, that is a normal fasting glucose. 0.5 to 1.5 milligram per deciliter is a creatinine. Cholesterol is around 200 mg per deciliter. Other you can learn. If you want to learn, you can learn. After that, we have seen the functions of blood. We have seen the different components of your plasma, right? So like this we have seen. Also I have also told you the WBC, RBC and platelets in the blood. We have seen the functions of RBC. We have seen about the how RBCs are made in the bone marrow. We have also seen about the hemoglobin. Remember, 1 molecule of hemoglobin will carry 4 oxygens because you have 4 globin chains in the hemoglobin structure and 4 heme units. Also because of the valency of that ferrous. Okay. Now we have seen hematocrit. Hematocrit means what? So hematocrit less than normal leads to anemia, more than normal leads to polycythemia. We have also seen how RBCs are made, what are the vitamins required for the maturation of RBC. Yesterday I was given a task to you. What are the synonyms of vitamin B12? You have to answer in the comment box. Yesterday मैंने जो आपको टास्क दिया था, मैंने जो आपको होमवर्क दिया था, तो होमवर्क क्या था? What are the 4 different synonyms of vitamin B12? आपने इसने भी कमेंट बॉक्स में बोला नहीं है, ठीक है? We have seen how RBC are made, how they are destroyed, right? We have also seen the negative feedback mechanism. के ऊपर भी क्वेश्चन आ चुका है. Erythropoietin is a hormone that produces the RBC in the blood and that regulate the production of RBC by negative feedback mechanism. You have to keep in mind. Okay. After that, we have seen the blood groups, right? So I would like to revise you once in a short blood groups. So if a person is having A antigen, if person is having A antigen on the surface of RBC, so that person will have A blood group. If a person has a antigen on the surface of RBC, the blood group will be A. And if a person has a antigen on a surface of RBC, so in A antigen, so that RBC will produce a anti-B antibody, anti-B antibody, anti-B antibody. Now, person having blood group A can donate a blood to another person having A or AB blood group. So, as a donor, compatible with A and AB. As a recipient, he is compatible with A and O. Okay? Because as a donor, अगर A वाले ने A को ब्लड दे दिया, इसके ऊपर जो एंटीजन है A, उसके ऊपर भी एंटीजन A रहेगा, तो एंटीजन एंटीजन रिएक्शन नहीं होता है ना. AB में भी एंटीजन क्या है, बोथ एंटीजन A भी है और B भी है, तो वहाँ पे भी इनकॉम्पेटिबिलिटी का कोई इशू नहीं है. तो ब्लड ग्रुप A पर्सन can donate a blood to another person having blood group A or AB, and he can receive a blood from another person having blood group A or O. Now, what do you mean by blood group B? What do you mean by blood group B? So, a person is having B antigen. A person is having B antigen on the surface of RBC, right? So then we call it as a blood group B, right? Blood group B. Now, person having blood group B, they has a B antigen on the RBC surface and they produces the anti-A antibody. Antibody कौन से बनाएगा, anti-A, right? So blood group B जिसका है, he is able to donate the blood to the another person having blood group B or AB, but he is able to receive blood from only person having blood group B and O. Now AB means what? So if a person has a both antigen A and B antigen on the surface of RBC, the blood group is known as a AB. Now AB is able to donate the blood only to the AB, but AB is able to receive the blood from everybody. So blood group AB is known as a universal recipient. Now O blood group. O blood group मतलब क्या होता है? So O blood group means a person having no antigen. There is no antigen on the surface of RBC, but that blood group will produce both antibody against A and as well as B. जिनका ब्लड ग्रुप O है, उसके सरफेस के ऊपर RBC के कोई भी एंटीजन नहीं होता, बट एंटीबॉडी दोनों बनाता है, एंटी A as well as एंटी B. So blood group having O, person having blood group O, so he is known as a universal donor. So he can donate a blood to anybody, but only receive blood from person having O blood group. So yesterday we have seen this. Now different type of WBCs and their functions. Different type of WBCs and their function. So as yesterday I have told you, all the blood cells are made from one pluripotent stem cell. So in the introduction part, WBC are of two type based upon the whether they have granules in their structure or not. They are classified into two type, granulocytes and agranulocytes. So WBCs are also known as leukocytes. Remember, WBCs are also known as leukocyte. There are two types. WBC के दो type होते हैं. There are main two types. So first type is nothing but the granulocytes, and second one is the agranulocytes. Those WBC having granules in their cytoplasm. जिसके cytoplasm में granules होंगे उसको granulocyte बोलते हैं. जिसके cytoplasm में granules नहीं होंगे उनको agranulocyte बोलते हैं. तो granulocyte कितने हैं? There are three granulocytes: neutrophils, eosinophils and basophils. All they are the granulocytes because they are having granules in their structure and they are known as a polymorphonuclear leukocytes. They are known as a polymorphonuclear leukocytes. agranulocytes, in that you have monocytes and lymphocytes. देखो आरबीसी हो गया अपना, डब्ल्यूबीसी मीन्स ल्यूकोसाइट, देयर मेन रोल इज नथिंग बट द डिफेंस सिस्टम ऑफ योर बॉडी. They are having role in the defense system of your body, and their WBCs are classified into two types, right? Granulocyte and agranulocytes. Granulocytes are neutrophil, basophil, eosinophil. Agranulocytes are nothing but the monocyte and lymphocyte. Sometimes they will ask you question. आपको क्वेश्चन आएगा कि one of the following cell is not a granulocyte. Yeah. So suppose for example they ask you question like following WBC is not a granulocyte, and they will give you option like neutrophil, basophil, eosinophil and monocytes. So monocyte is not a granulocyte. Monocyte, lymphocyte, they are a granulocyte. Now WBCs, they are the largest blood cells. You know, WBC are the largest blood cell, and as compared to RBC, but their count is very less. They count only 1% of blood volume. Only 1% of blood volume. ब्लड में ओनली 1% रहता है. तो दो दे आर द लार्ज ब्लड सेल्स बट देयर काउंट इज वेरी लेस, एंड दे आर हैविंग इंपॉर्टेंट फंक्शन इन द डिफेंस एंड इम्यूनिटी. जैसे कोई भी आपको बैक्टीरियल इन्वेजन होता है, सपोज यू गॉट अ बैक्टीरियल इन्फेक्शन हो गया, सो फर्स्ट सेल विच विल रिस्पॉन्ड टू दैट बैक्टीरियल इन्फेक्शन. कोई भी बैक्टीरियल इन्फेक्शन हो गया तो WBCs are the cells which can counter the bacteria. That's why WBC is having defense function. WBC is having defense function of your body. Now WBCs, they are classified, granulocytes, they are divided into three types: neutrophil, basophil, eosinophil, and they are divided on the basis of staining. You know, so we used to apply a stain on WBC, and some is a acidic stain, one is a acidic stain, and one is a basic stain. तो एक स्टेन होता है एसिडिक, एक होता है बेसिक, राइट? सो एसिडिक स्टेन जो है ना, सो एसिडिक स्टेन इज नथिंग बट द eosin. Eosin is the acidic stain that will stain a particular type of cell known as a eosin. Eosin is a acidic dye that will stain a particular type of cell known as a eosinophils. जो एसिड स्टेन है eosin, वो स्टेन करेगा पर्टिकुलर टाइप की सेल को, उसको बोलते हैं eosinophil. Now basic dye is there, that is known as a methylene blue. Methylene blue is a basic dye, and basic dye will stain a particular type of cell known as a basophils. So basophils, they are stained by basic dye. Eosinophils, they are stained by acidic dye, and neutrophils means what? So as the name indicates, उसके नाम में ही रखा है neutrophils. So they are neutral to both acid and base. Neutrophils, they will act as a neutral for acid as well as base, right? Eosinophils, they are stained by acidic dye. Basophils, they are stained by basic dye. Neutrophils, they are stained by both dye. Now what is the lifespan of neutrophil? देखो यहाँ पे न्यूट्रोफिल का लाइफ स्पैन क्या है? 2 to 5 days. न्यूट्रोफिल का लाइफ टाइम क्या है? 2 to 5 days. लाइफ स्पैन ऑफ न्यूट्रोफिल्स 2 to 5 days, and they are stained by both eosin as well as methylene blue. They get stained as a purple color. तो पर्पल कलर का आपको ये स्टेन मिलेगा यहाँ पे, राइट? नाउ eosinophils, what is life span? 7 to 12 days. They are stained by eosin which is acidic dye, है ना? eosinophil acidic dye से स्टेन होता है ना, eosin क्या है? acidic dye. That's why eosinophils are also known as acidophils. Remember, eosinophils are also known as acidophils. क्यों? Because eosinophils, they are stained by acid dye known as a eosin. Eosin is acidic in nature, and that's why eosin will stain a particular type of WBC known as eosinophils. Now methylene blue is a basic dye that will stain a particular type of cell known as a basophil, and neutrophils, they are stained by both dye, है ना? methylene blue क्या है? basic dye होता है ये, and this one is a basic dye, this one is a acidic dye. Acidic dye will stain a eosinophil. No, that's why they are known as acidophils. Basic dye will stain a basophil. That's why they are known as basophils. And neutrophils, they are stained by both dye. That's why neutrophils. Okay? So neutrophil, basophil, eosinophil, they are differentiated. They are classified. They are divided on the basis of staining technique. Neutrophils are stained by both dye. Lifespan कितना है 2 to 5 days. Okay, they will appear as a purple color. Now eosinophil, they will get a eosin dye. Lifespan is a 7 to 12 days. Eosin is a acidic type of dye. That's why eosinophils are known as a acidophils also. Basophils, because they are stained by basic dye, known as a methylene blue. Methylene blue, right? Lifespan is a 12 to 15 days. Now इसके ऊपर यहाँ पे एक क्वेश्चन ऑलरेडी आ चुका है. इन द ग्रैनुल्स ऑफ बेसोफिल, ऑल दे आर अ ग्रैनुलोसाइट. क्या है? यूसिनोफिल, न्यूट्रोफिल, बेसोफिल, ये क्या है? ग्रैनुलोसाइट है. यहाँ पे देखो, न्यूट्रोफिल्स, बेसोफिल, यूसिनोफिल, दे आर द टाइप ऑफ ग्रैनुलोसाइट्स. So they will have granules in their structure. They will have granules in their structure. As I say, granules. Now in the granules of WBC, known as a basophil, so basophils के granules में you have inflammatory chemicals. You have inflammatory chemicals. So in the granules of basophil, you have two compounds. First one is a histamine, and second one is a heparin. तो WBC जो बेसोफिल होते हैं, बेसोफिल के जो ग्रैनुल्स होते हैं, इन द बेसोफिल स्ट्रक्चर यू हैव हिपेरिन एंड यू हैव हिस्टामाइन, है ना. सो रिमेंबर, इसके ऊपर 2-3 बार क्वेश्चन आ चुका है. इन द ग्रैनुल्स ऑफ बेसोफिल्स, हिस्टामाइन इज स्टोर्ड अलॉन्ग विद द हिपेरिन. हिस्टामाइन एंड हिपेरिन आर प्रेजेंट इन द बेसोफिल ग्रैनुल्स. नाउ मोनोसाइट जो है, जिसका लाइफ स्पैन है 2 to 5 days, लिम्फोसाइट का 0.5 to 1 days, राइट. तो ऑन एन एवरेज लाइफ स्पैन ऑफ WBC is 7 to 12 days. 7 to 12 days. Now platelets means thrombocytes. They are also known as thrombocytes. And what is the role? They are involved in the blood clotting. There is a aggregation of platelet and there is a formation of platelet plug. That is the first step in the blood clotting. So they are involved in the cessation of blood or stopping of blood. Lifespan है उनका 8 to 11 days. Now you can see न्यूट्रोफिल का स्ट्रक्चर कुछ ऐसे होता है. न्यूट्रोफिल will look like this. So neutrophils, they will get stained and you will have this staining, you know. Neutrophils, they are neutral to both dye, right? So this is a nucleus known as a multi-lobed nucleus, and the neutrophils will have granules, so fine granules with the eosin and methylene blue stain, right? So in the granules of, you know, न्यूट्रोफिल के ग्रैनुल्स में क्या होता है? इन द ग्रैनुल्स ऑफ न्यूट्रोफिल यू हैव लाइसोजाइम. इन द ग्रैनुल्स ऑफ न्यूट्रोफिल यू हैव लाइसोजाइम. लाइसोजाइम क्या है? इट इज अ कलेक्शन ऑफ एंजाइम. एंजाइम कौन से एंजाइम? विल हैव हियर प्रोटीएज एंजाइम, then lipase, then nuclease, and carbohydrate metabolizing enzyme. So in the granules of neutrophils you have lysozyme, which is known as a collection of enzyme. In that you have different enzyme like protease, which will break down the protein, lipase will break down the lipid, nuclease will break down the nucleic acid, and carbohydrate digesting enzyme will break down the carbohydrates. Okay, now eosinophils to look like this. So they are known as a bilobed nucleus. तो देखो न्यूट्रोफिल्स में जो न्यूक्लियस है उसके 2-4 लोब्स होते हैं, मतलब, है ना. दिस इज वन लोब, दिस इज वन लोब, है ना. ये एक है. दैट्स व्हाई दे आर नोन एज अ मल्टी-लोब्ड न्यूक्लियस. न्यूट्रोफिल्स हैविंग मल्टी-लोब्ड न्यूक्लियस, यूसिनोफिल इज हैविंग बाय-लोब्ड न्यूक्लियस, मतलब 2 ही न्यूक्लियस होते हैं यहाँ पे. these two. Basophils means they are stained by basic dye, so they are also bilobed nucleus, you know. Then monocytes, they and a nucleus single lobe, okay, only one nucleus is there but pushed at one side, and lymphocytes, again nucleus occupying the whole cytoplasm. So these WBCs, they can be differentiated also on the basis of their appearance. You know, so a WBC on the basis of their appearance they can be differentiated. जैसे न्यूट्रोफिल में क्या है, in the neutrophil you have मल्टी लोब्ड न्यूक्लियस, इसमें जो न्यूक्लियस होगा दैट इज अ मल्टी लोब्ड न्यूक्लियस. In eosinophil you have bilobed nucleus. In the basophil again you have bilobed nucleus. In the monocytes, so a nucleus is a single one lobe nucleus or monolobe nucleus, but nucleus will be pushed at one side. Most of the part of nucleus is at on this side. Now in the lymphocytes, nucleus will occupy whole cytoplasm. How you can differentiate between monocyte and lymphocyte? मोनोसाइट में क्या है कि जो न्यूक्लियस होता है इट विल बी tilted at one side, एक साइड पे ज़्यादा होगा, and in the lymphocytes the whole cytoplasm will be occupied with the nucleus. Okay, now you can see leukocytes or WBC, they are known as the leukocytes or WBC. So what is the main function of leukocytes or WBC in the body? So they are having defense and immunity function. Defense and immunity, they are the first line defense of your body. तो जैसे ही कोई बैक्टीरिया, जैसे कोई वायरस, माइक्रोबियल ऑर्गेनिज्म आपके बॉडी में एंटर करता है, तो न्यूट्रोफिल्स, बेसोफिल्स, यूसिनोफिल्स विल गो एट इन्फेक्शन साइट, they will destroy the bacteria. Okay, the process of movement of these WBC is known as a chemotaxis. The process of movement of WBC is known as a chemotaxis, है ना. तो they have defense and immunity function, they will destroy the bacteria or antigen. WBC will have nucleus and some of them have granules in the cytoplasm. These granulocytes, they will have granules in the cytoplasm. That's why granulocytes known as a polymorphonuclear leukocytes. In that you have neutrophil, basophil, eosinophil. And A granulocyte में क्या है? A granulocytes, in that you have monocyte and lymphocyte. Now first is the neutrophils. पहला है neutrophils. So they are the active scavengers. क्या है वो? Active scavengers. Scavengers मतलब क्या? Active scavengers means they will catch the bacteria. They are the active scavengers of bacterial invasion. They will catch the bacteria as soon as they will you will have any infection in the blood in our body. The neutrophils will catch the bacteria, they will engulf the bacteria, they will destroy the bacteria, because in the granules of neutrophils, you know, in the granules of neutrophils what you have lysozyme. What is the lysozyme? इसके ऊपर क्वेश्चन भी ऑलरेडी आ चुका है ना. In the neutrophil granules, in the granules of neutrophil you have lysozyme. So lysozyme, what is lysozyme? It is a collection of enzymes like protease. You have protease enzyme which will digest the protein. You have lipase enzyme which will break down the lipids. You know, we have nuclease enzyme, you know, which will break down the nucleic acid, and carbohydrate metabolizing enzyme. So collection of enzyme known as a lysozyme, that is present in the neutrophil granules, which will helps the neutrophil to destroy the bacteria, and the process is known as a phagocytosis ना. मतलब क्या, न्यूट्रोफिल्स जैसे ही बैक्टीरिया को देखते हैं, बैक्टीरिया को देख के उसके पास जाके उसको एनगल्प करते हैं, है ना, खा लेते हैं, डिस्ट्रॉय कर लेते हैं बाय यूजिंग लाइसोजाइम. द प्रोसेस इज नोन एज अ फैगोसाइटोसिस. क्या बोलते हैं उसको? फैगोसाइटोसिस. प्रोसेस इज नोन एज अ फैगोसाइटोसिस. And movement of neutrophils towards the infection is known as a chemotaxis. इसके ऊपर भी क्वेश्चन ऑलरेडी आ चुका है. Movement of neutrophils towards the infection site is known as a chemotaxis, or movement of all WBCs, movement of WBCs towards the inflammation site, movement of WBCs toward the inflammation site known as a chemotaxis. इसके ऊपर भी क्वेश्चन आ चुका है, राइट? सो रिमेंबर न्यूट्रोफिल्स आर दे आर phagocytic in nature, है ना? Are you getting my voice properly? Are you getting my voice properly? Are you getting whatever I am telling you? Are you understanding it or not? Second type of WBC is a basophil. Now in the basophil there is a granules known as a vacuoles. In the basophil there is a granules known as a vacuoles, and they are having these chemicals, histamine, heparin. So in the granules of basophil you have histamine and heparin and other chemicals that will produce a inflammation. So basophil, they are the inflammatory cell basically. So basophil causes the inflammation, neutrophil causes the phagocytosis. Now eosinophils, they are also phagocytic in nature. They are also phagocytic in nature, but they are less active than neutrophils, and they will destroy big worms. They will destroy big worms, which cannot be easily phagocytized by neutrophils. They promote the inflammation by releasing the toxic chemical, but they can also decrease the inflammation by having. They also can decrease the inflammation because they have one enzyme known as a histaminase. मतलब neutrophils, they are phagocytic in nature, basophils, they are inflammatory in nature, and eosinophils, they are also phagocytic in nature. Now monocytes, they are the largest WBC and produces the chemical like interleukin 1, and which in turn increases the body temperature by acting on a hypothalamus in response to the microbial infection. जैसे आपको इन्फेक्शन होता है तो आपके बॉडी का टेम्परेचर बढ़ जाता है. क्यों बढ़ता है? Because once you got an infection, these WBCs will move towards the infection, and a neutrophil will destroy the bacteria, basophil will cause inflammation, and monocytes will cause a release of this interleukin 1 chemical, which increases the body temperature by acting on a hypothalamus. Stimulate the production of globulins by the liver. Globulins क्यों? क्योंकि globulins बनेंगे तो antibodies बनेंगे. It increases the production of T lymphocytes also. Now lymphocytes, they are smaller than monocytes, and they are found in the lymphatic tissue such as lymph nodes or spleen. T lymphocytes मतलब क्या? T lymphocytes, they are matured from the thymus gland. B lymphocyte, they will be matured from bone marrow only. ठीक है? तो आपको क्वेश्चन यहाँ पे कैसे आता है? यहाँ पे इसी स्लाइड के ऊपर अभी तक 7 या 8 क्वेश्चन पूछे, तो यू विल हैव एटलीस्ट 7 to 8 क्वेश्चंस ओवर हियर. ओके, तो जैसे फॉर एग्जांपल, व्हाट आर द टाइप्स ऑफ ग्रैनुलोसाइट्स? व्हाट आर द टाइप्स ऑफ ए ग्रैनुलोसाइट्स? टाइप्स ऑफ ग्रैनुलोसाइट्स, टाइप्स ऑफ ए ग्रैनुलोसाइट्स, इसके ऊपर क्वेश्चन आता है. तो व्हाट डू यू मीन बाय कीमोटैक्सिस? इसके ऊपर 2 बार क्वेश्चन आ चुका है. नाउ न्यूट्रोफिल्स आर phagocytic in nature, इसके ऊपर क्वेश्चन आ चुका है. And what helps the neutrophils to phagocytosize the bacteria? There is a lysozyme. What is lysozyme? There is a collection of enzyme having protease, nuclease, lipase and carbohydrate destroying enzymes. So इसके ऊपर भी क्वेश्चन 2 बार आ चुका है. What is there in a neutrophils granule? So it is a lysozyme. Now basophils, they produces the inflammation. Basophils, they produces the inflammation. इसके ऊपर क्वेश्चन आ चुका है. एंड व्हाट इज प्रेजेंट इन द बेसोफिल? इसके ऊपर 2-3 बार क्वेश्चन आ चुका है. जीपैट रिसेंटली आप देखो, जीपैट 2025 का पेपर, उसमें है क्वेश्चन, राइट? नाउ eosinophils, they are also phagocytic in nature. They are also phagocytic in nature, but they will phagocytosize a large worms. Monocytes, they will produce a chemical interleukin. तो इस टर्म, ऐसे कुछ टर्म्स है जो इसके ऊपर क्वेश्चन आते हैं आपको, राइट. नाउ lymphocytes, there are two lymphocytes, yeah, T lymphocytes and B lymphocytes. They are found in the lymphatic tissue. Now last one is a platelet. Platelet means what? They are also known as the thrombocytes. So they are involved in the hemostasis. They are involved in the coagulation of blood. Now the production is stimulated by hormone known as thrombopoietin. तो प्लेटलेट को कौन हार्मोन कंट्रोल करता है? थ्रोम्बोपोएटिन, राइट. डब्ल्यूबीसी को कौन करता है? डब्ल्यूबीसी का हार्मोन होता है आपका सीएसएफ. CSF. So RBC production is regulated by erythropoietin. Platelet production is regulated by thrombopoietin, and WBC production is regulated by CSF. What is the CSF? It is not a cerebrospinal fluid. What is that? It is a colony stimulating factor. CSF, colony stimulating factor. So there are different hormones like GCSF, GMCS. We'll discuss in upcoming part. Right? So you can see over here, what is the normal count of neutrophils? Neutrophil का normal count होता है 2.5 to 7.5 into 10 raised to 9 per liter, or 40 to 75%. Eosinophil 0.04 to 0.04 into 10 raised to 9 per liter, or 1 to 6%. Basophils less than 1%. Monocyte, lymphocyte, no, monocyte 2 to 10%, lymphocyte 20 to 50%, total 100%. Now basophil look like this after staining. ये basophil will look like this after staining. Neutrophil will look like this. Staining करने के बाद neutrophil ऐसे कुछ नजर आएंगे माइक्रोस्कोप के नीचे. Eosinophil will look like this after staining. Now this is known as a phagocytosis. Phagocytic action of neutrophil. Demonstration of phagocytic action of neutrophil. जैसे यहाँ पे this is a neutrophil and this is a neutrophil cell. न्यूट्रोफिल नाम में क्या रखा है? बोलते हो नाम में क्या रखा है? नाम में रखा है न्यूट्रोफिल. न्यूट्रो मतलब न्यूट्रलाइजेशन. तो इट विल न्यूट्रलाइज द वर्म्स, इट विल न्यूट्रलाइज द माइक्रोब्स. न्यूट्रोफिल. So this is a neutrophil cell. Suppose this is a microbial infection. So neutrophil will go at infection site and it will form a pseudopodium. Pseudopodium means it will get attracted towards the microbial cells. Pseudopodium will form a mouth. So they will engulf the bacteria. Bacteria will be engulfed and they will be destroyed, known as a phagocytosis. So what are the components of plasma? Plasma is made from 92 to 93% water. तो प्लाज्मा है उसमें 92 to 93% क्या रहेगा? वाटर. बट रिमेनिंग इज अ सॉलिड एंड गैसेस. What are the solid content of your plasma? In the plasma you have organic substances, like example plasma proteins. So albumin is there, globulin is there, then blood clotting factors are there, fibrinogen and other, right? Then you have amino acids. They may be either essential amino acids or non-essential amino acids. You have carbohydrate in the blood, plasma, glucose. Lipids are there like triglyceride, cholesterol, phospholipids. You have some hormones, internal secretions. There are some inorganic content, inorganic substances like sodium, potassium, calcium, magnesium, bicarbonate, chloride, phosphate, iodide, iron and copper. These are the inorganic content of your plasma. So you have gases in the plasma also. So for example, oxygen, carbon dioxide, nitrogen. You have different enzymes in the plasma. Different enzyme: amylase, carbonic anhydrase, acid phosphatase, alkaline phosphatase, lipase, protease, transaminase. You have non-protein nitrogenous substances like ammonia, waste product. तो अपन बोलते हैं waste product. Urea, uric acid, xanthine, creatinine, ammonia. So these are the component of plasma. Okay. Now sometime they ask you the pH of body fluids. देखो बहुत बार questions already they have already asked questions from the pH of different body fluids. So I have listed the pH over here, but what is most important, so they can ask you pH, rather they have already asked what is the pH of gastric juice, body fluids, different body fluids and their pH. तो इसके ऊपर ऑलरेडी आ चुका है. What is the gastric juice pH? What is the pH of stomach? So that is a 1.2 to 3, or you can say 1 to 3. So,
हेलो.
हाँ
गुड इवनिंग सर.
कौन है, कौन बोल रहे हैं?
माय नंबर
10 मिनट को तो मिलेगी. pH of gastric juice or stomach, it is a 1 to 3. Then pH of vaginal fluid around 3.5 to 4.5. And a vaginal fluid pH, they can ask pH of vaginal fluid, okay, 3.5 to 4.5. So vaginal fluid pH is acidic in nature. यहाँ पे याद रखना है acidic. Now ये acidic क्यों होता है? Why the pH of vagina is acidic? Because in the vaginal cavity you have lactobacilli, lactobacillus vaginalis. So you have lactobacillus vaginalis that will maintain a pH of vagina around 3.5 to 4.5. pH of gastric juice is important. इसके ऊपर ऑलरेडी क्वेश्चन आ चुके हैं. Acidic pH, very strong acid, 1 to 3. Then pH of CSF, cerebrospinal fluid का pH होता है आपका 7.4. pH of pancreas is a alkaline in nature. Pancreas होता है उसका pH alkaline होता है, and pancreatic pH is a 7.1 to 8.2. Then bile, in a bile gallbladder you have bile juice, you know, that is again alkaline, 7.6 to 8.6. Then pH of urine, बहुत बार पूछा है pH of urine, 4.6 to 8. pH of saliva पूछा है 6.35 to 6.85. pH of milk 6.8. डिस्टिल वाटर का pH 7 होता है, ये भी पूछा है एक बार. What is the pH of blood? 7.35 to 7.45. So it is a slightly alkaline. pH of semen 7.2, 7.6. Then pH of tears 6.5 to 7.6. So rest of the pH I have listed over here, but वो ज़्यादा कुछ पूछेंगे नहीं आपको, है ना. लेमन जूस इसका कोई नहीं पूछता. pH of soft drinks 10, but most of the body fluid pH they can ask you. That's why I have included this slide in the lecture. Okay, so pH of different body fluids. Even nowadays you have AIs in your mobile, so you can ask to the AI, give me the different body fluids and their pH. See, there are different ways to study, but there is no alternative for study. Now in the plasma you have blood clotting factors. Now this chart is very important, and in the plasma, as I told you, you have different plasma proteins and different blood clotting factors. So what are the components of plasma? Different plasma proteins and blood clotting factors. So there are, there are around 16 blood clotting factors. There are around 16. So total blood clotting factor कितने हैं? There are 16 blood clotting factors. There are 16 blood clotting factors. I will explain you in upcoming part also. But these blood clotting factors are very important, and a lot of times they already ask question on this. And you can expect at least within this slide they have already asked questions at least 10 times, 10 to 15 times. So how many blood clotting factors are there? 16 are there. Remember, I will explain you. I will tell you the name of that. Now what is blood clotting factor number 1? So what is blood clotting factors? Yeah, blood clotting factors, what are they? So they are nothing but the blood clotting factors are nothing but the proteins in nature. They are a protein. They are inactive glycoproteins. They are inactive glycoprotein. So These blood clotting factors are nothing but the inactive glycoprotein. They are a glycoprotein in the plasma or blood, and they are nothing but the first one is a fibrinogen. First one is a fibrinogen. First blood clotting factor is a fibrinogen. Second one is known as a prothrombin. Second blood clotting factor is known as a prothrombin. So, minute. There are 16 blood clotting factors, and on these blood clotting factors they have already asked questions multiple times, right? And what are the blood clotting factors? What are these blood clotting factors? I will explain you one by one, right? Now first one is a fibrinogen, second one is a prothrombin. Okay, remember, because multiple times they have already asked question on this. What are the different blood clotting factors? So there are 16 blood clotting factors in the plasma, and they are a inactive glycoprotein. Remember, क्या होता है inactive glycoprotein. So fact number 1 is known as a fibrinogen. Fact number 2 is known as a prothrombin. Blood clotting factor 3 is known as a tissue factor or thromboplastin. Blood clotting factor 4 is known as calcium or inorganic calcium. 5 is known as a labile factor or proaxalrin or AC globulin. Fact number 7, stable factor or proconvertin. Fact number 8 is known as a anti-hemophilic factor A. Anti-hemophilic factor A. Anti-hemophilic globulin or anti-hemophilic factor A. Factor number 9 is known as a Christmas factor or plasma thromboplastin component or anti-hemophilic factor B. Factor number 10 is known as Stewart power factor. Factor number 11 is known as a plasma thromboplastin antecedent component PTA, also known as anti-hemophilic factor C. Factor number 12 is known as a Hagman factor. Factor number 12 क्या है? Hagman factor, also known as a contact factor, also known as a glass factor. Fact number 13 is known as a fibrin stability factor. So देखो, most of the books they have given only 13 blood clotting factors. Most of the books they have given only 13 blood clotting factors. Most of the books they have given only 13, but there are 16 blood clotting factors. There are 16. Okay, so first one is a fibrinogen, second one is a prothrombin, third one is a tissue factor or tissue thromboplastin, fourth one is known as a calcium, fifth one is known as a labile factor or proaxalrin or AC globulin. तो जो फिफ्थ वन है वो प्रोएक्सेलरिन है. तो सिक्स्थ है क्या? नहीं, मोस्ट ऑफ द बुक्स दे आर नॉट गिवन द सिक्स फैक्टर. Most of the books they are not given the fact number 6, but there is fact number 6. And what is fact number 6? It is an active factor of fact number 5. So fact number 5 क्या है? Proaxalrin. So what is fact number 6? It is a axalrin. Okay. Fact number 7 is a stable factor or proconvertin. Fact number 8 is a anti-hemophilic factor A. Fact number 9 is a Christmas factor or anti-hemophilic factor B. Factor number 10 is stored power factor. Factor number 11 is a anti-hemophilic factor C. Factor number 12 is a Hagman factor. Factor number 13 is a fibrin stability factor. Okay. Factor number 14 is there known as a pre-calcarein. Factor number 15 is there calcarein. Factor number 16 is there high molecular weight kinogen. Okay. But most of the books they have given only 13 blood clotting factors. But ठीक है, यहाँ पे देखो क्वेश्चन कैसे कैसे आया. So multiple times question they are already asked on this slide. So factor number 1 minimum 3 times. 3 times in the GPAT exam. Also in an Niper DI Pharmacist. Prothrombin at least 5 times. At least 5 times they ask. Factor number 2. At least 3 times. Calcium at least 4 times. Fact number 5 एक दो बार पूछा है 2 times. Fact number 7 once. Fact number 8 minimum 2 times. Fact number 9 minimum 4 times. Fact number 10 minimum 3 times. Fact number 11 once they already asked. Fact number 12 minimum 3 times. Number 4 fact number 13 minimum 4 times in the GPAT exam. Also in a Niper अगर 4 बार GPAT में पूछा है तो 1-2 बार 3 बार Niper में पूछा होगा. So multiple times, at least 20 times they have already asked question on this. And recently from last few years they are asking question like this. Match the following blood clotting factor number with their respective name. है ना, ऐसे भी 3-4 बार क्वेश्चन आ चुके हैं. So very important. And again in the GPAT 2022 as well as GPAT 2018, what are the vitamin K dependent blood clotting factors? What are the vitamin K dependent blood clotting factors? Remember इसके ऊपर multiple times पूछा है vitamin K dependent blood clotting factors. What are these? Fact number 2 and fact number 7, 9 and 10. है ना, यहाँ पे देखो these two are odd, these two are even. Fact number 2, 7, 9 and 10. 2 are odd, 2 are even. So these are the vitamin K dependent blood clotting factors. So it means only there is a one chart or only there is a one list known as the blood clotting factors. And on that chart, on that list, they have already asked question multiple times and a minimum 20 times. So I will explain you what are the 16 blood clotting factors. Don't worry in the coming slides. Right now, the process of blood coagulation. What are these blood clotting factors? Blood clotting factors are nothing but the inactive glycoprotein in the ये proteins inactive proteins in the plasma. Inactive है, they needs to be activated, and some of them need a vitamin K for activation. Some of them needs a vitamin K for activation, and that's why they are known as vitamin K dependent blood clotting factors. So these blood clotting factors, once they are activated, so then the blood clotting will start. Okay, now the coagulation of blood known as a hemostasis. The blood coagulation is known as a hemostasis. Coagulation of blood is known as a hemostasis, also known as a cessation or stopping of blood. Blood coagulation is known as a hemostasis. Now blood coagulation should be there. Blood coagulate होना चाहिए कब? जब आपको इंजरी हो, जब आपका वेन कट जाए. If there is injury to your tissue, if there is injury to your blood vessel, then to prevent the blood loss, blood should stop. There should be clotting of blood, है ना. That is a physiological in nature. अगर आपका वेन कट होता है ना, ब्लीडिंग हो गया तो आप मर जाओगे ना. तो ब्लीडिंग रोकना चाहिए तो उसके लिए ब्लड क्लॉटिंग मैकेनिज्म होता है ना, दैट इज नोन एज कोएगुलेशन ऑफ ब्लड. बट it should be clotting of blood should be when there is injury to your blood vessel or whenever there is injury to your tissue, then only blood clotting should be there. But without injury blood clotting if occurs, that is known as a pathological. तो ब्लड क्लॉटिंग इंजुरी के बाद ही होना चाहिए है ना. पहले ही अगर हुआ ब्लड वेसल में ब्लड क्लॉट होगा तो उसको बोलते हैं थ्रोम्बस और दैट इज अ पैथोलॉजिकल. नाउ if you don't know how blood coagulation occurs, you know, how you can understand the anticoagulant and all that. So blood coagulation is a cascade of series of activation and of blood clotting factors. So blood coagulation, there are two stages. There are three stages of blood coagulation: stage 1, stage 2, stage 3. So complete blood clotting occurs in the three stages: stage 1, stage 2, stage 3. There are two pathways for blood coagulation. Blood clotting के लिए दो pathway होता है. So first pathway is known as the intrinsic pathway. First one is known as the intrinsic pathway. So as the name indicate, intrinsic मतलब क्या है? So that occurs inside the blood, inside the body. So inside the body intrinsic pathway occurs and outside the body extrinsic pathway occurs. है ना. So suppose if there is a है ना से blood vessel है आपका, there is a blood vessel. So inside the blood vessel, inner layer of blood vessel is smooth, known as a endothelium. बट ड्यू टू एनी रीजन जैसे डिपोजिशन ऑफ कोलेस्ट्रॉल या फिर एनी एंडोथेलियल लेयर्स आपका खराब हो गए, तो डैमेज टू द एंडोथेलियल लेयर एंड द एक्सपोजर ऑफ कोलेजन एक्सपोज होगा. एंडोथेलियल डैमेज अगर खुरदुरा हुआ है, नॉर्मली एंडोथेलियल लेयर ऑफ योर ब्लड वेसल इज स्मूथ, नाउ इट बिकम्स रफ और रफ. so then what will happen, then there is a release of two factors known as a kallikrein and high molecular weight kinogen. Kallikrein and high molecular weight kinogen. So these are also clotting factors, which is a clotting factor number 14, 15 and 16. Now so once there is a endothelial damage, exposure of collagen, what will happen, there is a, and as I told you, blood clotting factors are inactive in the blood. All these blood clotting factors, they are inactive in the blood. They needs to be activated, and the activation process is known as a cascade of blood clotting factors. So once there is a damage to your blood vessel, endothelial layer, smooth will be converted into rough, and that will leads to exposure of collagen. Okay, and then there is a release of these two factors, high molecular weight kinogen and kallikrein. So that two factor will convert inactive blood clotting factor number 12 to the active factor. ये जो लिखा है उसके आगे, that is nothing but the active factor. So inactive factor will be converted into active, and factor number 12, that is Hagman factor, that is converted into factor number 12A, active factor. Now again, along with the kinogen, inactive factor 9, Christmas factor, will be converted into active Christmas factor. Now active Christmas factor along with the calcium convert inactive, you know, sorry, anti-inflammatory factor C. Factor number 11 is anti-inflammatory factor C. Now active factor 11 will convert inactive Christmas factor to the active, which will convert inactive Christmas factor converted into active. Now active Christmas factor along with the factor number 8 and calcium convert factor number 10 steward power factor into active steward power factor. And again, ये जो exposure collagen होता है, collagen exposure, that leads to aggregation of platelets, and platelet aggregation leads to release of phospholipids, and these two are also stimulate, this, so factor number 10 along with the factor number 5, they convert inactive thrombin So they convert, they stimulate the prothrombin activator. एक कॉम्प्लेक्स जो है उसको बोलते हैं प्रोथ्रोम्बिन एक्टिवेटर. प्रोथ्रोम्बिन कन्वर्ट थ्रोम्बिन इनटू ना, प्रोथ्रोम्बिन कन्वर्ट ना प्रोथ्रोम्बिन इनटू थ्रोम्बिन. और थ्रोम्बिन कन्वर्ट इनएक्टिव और है ना soluble fibrinogen to the insoluble fibrinogen, and that causes the formation of fibrin threads. Then inactive fibrinogen will be polymerized from a fibrin threads along with the calcium and factor number 13 known as the fibrin stability factor. There is a formation of blood clot. So this is known as a intrinsic pathway. This is known as a intrinsic pathway. जैसे आपके ब्लड वेसल को डैमेज होता है तो प्री कैलिकाइरिन हाई मोलिकुलर वेट काइनेजन विल कन्वर्ट इनएक्टिव फैक्टर नंबर 12 एगमन फैक्टर टू एक्टिव फैक्टर 12. देन एक्टिव फैक्टर 12 along with the high molecular kinesin convert active factor inactive 11 factor to the active 11. Then active 9 to the inactive 9 to the 9 to 9. Then inactive 10 to the active 10, which activate the prothrombin activator. प्रोथ्रोम्बिन जैसे एक्टिवेटर हो गया तो इट विल प्रोथ्रोम्बिन एक्टिवेटर कन्वर्ट प्रोथ्रोम्बिन फैक्टर नंबर 2 इनटू द थ्रोम्बिन. थ्रोम्बिन का काम है कन्वर्जन ऑफ फाइब्रिनोजन, दैट इज फैक्टर नंबर 2, दैट इज फैक्टर नंबर 1. इनएक्टिव फाइब्रिनोजन ये होता है सोल्यूबल. नाउ इट इज सोल्यूबल इन द प्लाज्मा, बट सोल्यूबल फाइब्रिनोजन इज कन्वर्टेड इनटू इनसोल्यूबल फाइब्रिनोजन. Now that will be polymerized and converted into fibrin threads. Fibrin threads along with the calcium and factor number 8 that is converted into fibrin or blood clot. This is known as a intrinsic pathway. So ये जो prothrombin activator है, up to that there is a stage 1. Okay, up to the prothrombin activator stage 1. So prothrombin will convert prothrombin into thrombin, that is stage 2, and then in conversion of soluble fibrinogen to insoluble fibrin, that is stage 3. Now extrinsic pathway, suppose if you are tissue damage, आपको अगर आपसे बाइक से गिर गए आप तो जब ब्लीडिंग होता है कैसे होता है, तो वंस यू गॉट टिश्यू ट्रामा, आपको कुछ लगा है, दैट इज अ टिश्यू ट्रामा, एंड टिश्यू ट्रामा लीड्स टू रिलीज ऑफ टिश्यू थ्रोम्बोप्लास्टिन फैक्टर नंबर 4 जो है टिश्यू फैक्टर. देन टिश्यू थ्रोम्बोप्लास्टिन convert glycoprotein and phospholipids, and factor number 7 along with that, and tissue thromboplastin factor number 7 and glycoprotein convert inactive factor number 10 toward power factor to active factor 10A. active मतलब A for active. Then active factor 10 along with the calcium and factor number 5 है ना, activate the prothrombin activator. प्रोथ्रोम्बिन एक्टिवेटर जब एक्टिवेट होता है तो कन्वर्ट थ्रोम्बिन इनटू, it will convert prothrombin into thrombin. Thrombin will convert inactive fibrinogen to active fibrinogen, and then this is known as a extrinsic pathway. Blood clotting occurs in a body. Blood clotting takes place by two pathway. First one is a intrinsic pathway. Intrinsic pathway matlab that occurs inside the blood vessel. Clotting that occurs inside the blood vessel. Occurs in the blood vessel, that is intrinsic. Extrinsic pathway means blood clotting that occurs due to the tissue damage. Tissue को trauma हो गया तो blood clotting. तो extrinsic pathway हो गए. Now what are the different plasma proteins in the plasma? जैसे अपन ने देखा है in the plasma you have different components. One of the components is the blood clotting factors. वो अपन ने देखा है. So another component is a plasma proteins. Another component is a plasma proteins. So very important again. A lot of times they already ask question on this. So plasma proteins comparatively, or on this slide they already asked question at least 8 times. इसी स्लाइड के ऊपर कम से कम 8 या 10 बार क्वेश्चन आ चुका है, मिनिमम 8 to 10, कम से कम 8 से लेकर 10 बार के ऊपर क्वेश्चन आ चुका है GPAT Niper DI Pharmacist. Now here I have table name of protein, molecular weight and functions, right? तो देखो पहला है आपका अल्बुमिन. तो अल्बुमिन is the most abundant protein in the plasma, most abundant protein in the plasma, and in a GPAT 2020 they have asked molecular weight. Now albumin के ऊपर ही बहुत सारे क्वेश्चंस है. If you search a term albumin, अगर आपने albumin term सर्च किया in the previous year question paper database, you will find at least 20 questions, at least 20 question on albumin. We'll discuss, है ना, don't worry. So albumin is the most abundant plasma protein, and the molecular weight of albumin is around 65,000 to 69,000 Dalton. This was a question in GPAT 2020. What is the molecular weight of albumin? Now what is the role of albumin? Albumin has a different role. तो पहला रोल है उसका ना, तो in a transportation, it will transport a hormones, ना, amino acids, vitamins and drugs. Transportation, it is a transport protein. Then also albumin regulate the pH of blood, one of the important buffer in the body. Also this albumin is responsible for the maintenance of osmotic pressure or oncotic pressure of your blood. तो albumin क्या करता है, osmotic pressure, oncotic pressure regulate करता है blood का. Also important buffer in the blood. Also it is known as a transport protein for endogenous chemicals and hormones. Also it is a binding site for drug, and after the absorption of drugs, some drugs depending on the physicochemical properties they will bind to the albumin. Is a binding site also, but वो बाद में देख लेंगे. But remember the function: transportation, regulation of pH, then act as a buffer, and then regulation of osmotic pressure of your blood. Now second one is a globulin. Second plasma protein is a globulin. Albumin क्या है? It is a, है ना, second one is a globulin, and it is a large protein. There are 3 types of globulin. इसके 3 टाइप होते हैं. There are 3 types of globulin. Molecular weight will range from 90,000 to 1.5 lakhs Dalton. There are 3 types of globulin. यहाँ पे 3 टाइप होते हैं: अल्फा ग्लोबुलिन, बीटा ग्लोबुलिन एंड गामा ग्लोबुलिन. Alpha, beta and gamma. There are 3 type of globulin: alpha, beta globulin, gamma globulin. Now alpha, beta globulin, they are a transport protein. No? Alpha, beta globulin, they are a transport protein. But gamma globulins, they are known as antibodies. Gamma globulins क्या है? एंटीबॉडीज. जैसे एंटीबॉडी जी, IgG, IgM, IgI, IgE, ये सब क्या है? gamma globulins है. And in the body the albumin globulin ratio is there. In the body albumin globulin ratio is maintained. And what is the albumin globulin ratio? इसके ऊपर भी क्वेश्चन आ चुका है एक बार. So in the body albumin globulin ratio is there, which is 2:1. Remember, in the body albumin globulin ratio is maintained. In the disease condition this ratio will change. तो albumin globulin ratio कितना होना चाहिए? 2:1. 2 part of globulin, 1 part of sorry, 2 part of albumin and 1 part of globulin. Now third one is a fibrinogen. Fibrinogen has a molecular weight of 3.4 lakhs to 5 lakhs. क्या फंक्शन है उसका? ब्लड क्लॉटिंग. नेक्स्ट प्रोटीन इज अल्फा 1 एसिड ग्लाइकोप्रोटीन. molecular weight is 44,000 dalton, and alpha 1 acid glycoprotein also known as orthomucoid. So alpha 1 glycoprotein जो है that is increased in the inflammation, increase in the acute inflammation. So that's why it is an indicator for acute inflammation. Orthomucoid है ना. Acid glycoprotein alpha 1 acid. Now alpha 1 globulin molecular weight is around 59,000 dalton. Alpha 2 globulin molecular weight 1.34 and 1.34 lakhs. Alpha 2 globulin is known as a ceruloplasmin. इसके ऊपर क्वेश्चन आ चुका है. Why it is known as ceruloplasmin? We are discussing the plasma proteins. Alpha 2 globulin is known as a ceruloplasmin. Why? Because alpha 2 globulin is a binding site for cupric ion. Alpha 2 globulin is known as a ceruloplasmin. क्यों बोलते हैं ceruloplasmin इसको? Because it is a binding site for copper ion. No, binding site for copper ion. That's why it's known as ceruloplasmin. You know. Also has one enzyme known as the peroxidase, enzyme responsible for the incorporation of iron into transferrin. Transferrin क्या है? Transferrin is known as the iron transport protein. है ना. Then lipoproteins होते हैं in the plasma ranging from 2 lakhs to 3.4 lakhs. है ना. Transportation of lipids are the function of lipoprotein. Hemoglobin and molecular weight is a 64.5 thousand dalton. What is the role of hemoglobin? To transport the oxygen. Then transportation is the role of hemoglobin. Right now, this is a different plasma proteins, their molecular weight and their role. इसके ऊपर ही कम से कम 10 बार क्वेश्चन आ चुका है. And this table we are again going to see in the general pharmacology. Don't worry about that. But अगर यही से ही आप अगर कैप्चर करते हो उसको, that will be a better. Remaining part we'll see the tomorrow. Okay, tomorrow we'll see the disease of the different diseases of the this blood. And so I hope you have understood. If you have understood, please write yes in the comment box. अगर आपको समझ में आया तो yes लिखिए comment box में. Okay. Tomorrow we'll see the remaining part and also we'll discuss some MCQs.